Rimpi, I can't hear you. What's the matter?
Am I audible now?
Yeah.
I just wanted to say thank you so much, everyone, and thank you, Dr. Rashmi Sharma, Professor Sunita Huda, Dr. Vineet Singh, and Dr. Sumit Sahani for chairing the session. Apologies for the technical errors over here. Moving over to the next session, while we are learning about how we can learn from the nature and teach our children, now we're moving over to the next talk. And for that, I would like to invite our coaches, Dr. Rahul Dev and Dr. Sushma Bhardwaj, Assistant Professor, Department of Zoology, Acharya Narendra Dev College. Please.
Thank you, Rimpi. It is our pleasure to introduce the first chair of the session, Professor Arijit Chaudhary, to our learned audience. Professor Chaudhary has been teaching for the past 16 years in the Department of Physics at Acharya Narendra Dev College, University of Delhi. He holds a PhD degree in experimental semiconducting thin film-based sensors and has postdoctoral work experience in Australian National University, Canberra, Australia. He has research collaborations with Joseph Stephen Institute, Ljubljana, Slovenia, and Norwegian Institute of Air Research, Keller, Norway. His research interest lies in the field of electronic nose for artificial olfaction, integrated semiconductor gas chemical chambers, quartz crystal microbalance sensors, ambient air pollution detection and mitigation, water purification, atomic layer deposition, and much more. He has 107 research publications to his credit in various national and international journals. He has completed 10 research projects, few with international collaborations with fundings from agencies including DBT, University of Delhi, Department of Information Technology. He has guided students for PhD research. Sir has been winner for the Best Innovation Idea Award from Delhi University Innovation Project in 2015 and recognized for teaching excellence and innovation by University of Delhi. Sir, on behalf of the Ciliate Biology family, I welcome you. I would also like to introduce our other chair for this session, Mr. Ravindra Kumar Sagar. Sir is an associate professor in the Department of Zoology at Acharya Narendra Dev College for the past 17 years. His area of research involves work on herbal extraction used in mitigating gamma radiated induced damage to hemopoietic system at Institute of Institute of Nuclear Medicine and Allied Science, DRDO. He has successfully completed one innovation project funded by University of Delhi and is presently co-investigating project entitled Attractive Toxic Sugar Bait to Control Mosquitoes in Different Regions of Delhi and NCR, funded by ICMR. Looks like an interesting project. He has 15 international and 3 national publications, a book chapter, and 2 patents to his credit. He is also contributing as convener and member of syllabus drafting committee for animal diversity and human physiology papers. We are glad to have you with us today, sir. Heartily welcome you. May I now request Professor Arijit Chaudhary to introduce our esteemed chairperson to the session, esteemed speakers to the session.
All right. Thank you, Dr. Bhardwaj, for those eloquent and kind words. I would like to invite, it's an honor and a privilege to be part of this committee and chairing a session in this esteemed conference. To the speaker, I would say that's Buongiorno, Dottoressa Valentina Serra. Dr. Serra is a researcher in biology department in Pisa University, Italy, where she has carried out her PhD on biodiversity of ciliates and their symbionts from Indian subcontinent. She has developed the idea of the next generation taxonomy and is actively contributed to writing those funded projects, including S2020 MSc Rise NGTX project, of which currently she is a project assistant also. She has worked in the field of ciliate biodiversity, systematic, and symbiosis for the last 10 years. So welcome, Sayonara, Valentina Serra.
Thank you. Thank you. Thank you for the introduction. Good morning to all, and thanks for the invite, and thanks to the organizer of this symposium. I'm really proud to be here, also because I had the chance and the privilege to work in India during my PhD. So for me, it's a particularly important to be here. So I'm going to share my screen. Oh, it works. Okay. Can you hear me and see my screen?
Yes, ma'am, loud and clear.
Okay.
Yes, please.
Fine, you can see the first slide. Okay,
ma'am.
so I'm gonna start. Okay, so I'm really proud and really happy to share with you this presentation that deal with one ongoing project funded by the European Commission. The title of the project is Next Generation Taxonomy, Ciliophora and their Bacterial Symbionts as a Proof of Concept. The acronym of the project and is NGTAX. So today I'm gonna just to briefly describe our project and the rationale behind and the practical applications. Our project is Horizon 20. It was founded in the frame of Horizon 2020 and is a RISE project, so a research and innovation staff exchange. That it means that funds short-term exchange of personnel. That means mostly young researchers, but also experienced researchers, technical and administrative and managerial staff between different academies, but mainly among universities and companies, because one of the main goals of this project is just to create connections between the university and company universe, just to introduce also researchers to this really interesting field, so the field in which they can work in a company. So our project was founded in 2020, but for the pandemic, we had to postpone the starting date to the June of 2021, and now we are requesting an extension to the European Commission, and we hope to end the project in 2026. Our project, you will see, deal with the taxonomy of ciliates, and more broadly, we hope also taxonomy of other organisms, just introducing novelty in the descriptors of organisms, but we will deep this thing later. So our project at the moment has as partners and units members of our network 8 different universities from Europe, from Asia, and from Africa. More precisely, we have University of Pisa, that is the unit coordinator. Then we have Universitat Innsbruck and the Max Planck Institute in Tübingen and Wuppertal University from Europe. And then in Asia, we have as partner the Austrian University of China, and in Africa we have Addis Ababa University from Ethiopia, Egerton University from Kenya, and Free State University in Bloemfontein from South Africa. We have also 3 different companies. One is the sequencing companies from Germany, Genevitz. One is a wastewater treatment plant in Italy, with which we collaborate just also for sampling and study the biodiversity of ciliates in this kind of particular environment. And then we have also Palustrin Design Oy from Finland, which is a company which deal with repristinate freshwater environments in like say compromised habitats. We are proud to say that coming soon, we could have 3 more partners: University of Pavia and from Italy, Giant Genome Institute from USA, and also another company that deal with microscopy, optic, and photography. We hope to have them soon as new partners. Our project is quite ambitious. We want to, let's say, perform to 277 person months. It means that we want people just come around our network at least 277 months. We hope to achieve this goal, to encourage people mobility and secondments, and just to exchange their knowledge, and to make a new experience that can boost their career, especially for young researchers, of course. Of course, this project really help this kind of mobility, providing a kind of fee that shall include travel and accommodation costs for people who move, is around 2,100 euro per month. And with that, the project wants to help people just move and make a new experience in the field of our project. So in brief, the aims of our project are several, but mainly to summarize, NGTax project want to propose and promote a new advanced standard for taxonomic description of organisms with that workflow that we call next generation taxonomy, and we will explain later. Of course, we want to build an international, intersectoral, and multidisciplinary network, and we are trying to do that, and hopefully create long-lasting collaborations with our partners, and maybe during the course of the time also finding new ones. And one of our main goal is for sure to boost careers of young researchers, making them move, have new experience, and experience new field and new working environments. So, but now just go through the rationale behind our project. So basically what our project deal with. And for that, we can just start from the definition of taxonomy. Taxonomy, as you may know, is a very important discipline. It's the science of defining and naming groups of biological organisms based on shared characteristics, and more recently based on evolutionary relationships. To become a really good traditional taxonomy, it means you have to spend a lot of years of study, of working on the field, and just to become a good traditional taxonomist on maybe also restricted groups of organisms, it means that you have really to work a lot and work so much. For this reason, and maybe also for the rising of new molecular techniques around the 80, 90 of the last century, the traditional taxonomy faced a little bit of crisis. But fortunately, at the beginning of the 2000s, with the introduction of new methods, we could have that so-called the integrative taxonomy, which mixed traditional taxonomy methods with ultrastructural analysis and, of course, molecular studies. But now, at a certain point, we wonder, it is maybe time for a step forward. I mean, the taxonomy can just be enriched by new techniques and also new concepts. So we look around, and nowadays we realize that actually a lot of new tools, new powerful tools arose from a technical point of view. I'm just speaking about genomics, bioinformatics, and all these and novel sequencing methods that are for sure new powerful tools that maybe can be part of the new concept of taxonomy. And in particular, we thought about the possibility to use the whole mitochondrial genome or the mitogenome as a new, very useful molecular marker to add to the description of one organism. Why we thought about the mitochondrial genome? Well, because mitochondrial genes are already employed for species discrimination and description, especially if they are cryptic. But let me say that working in this field, I know very well that sometimes to get this kind of genes just by the PCR methods and PCR approach is not so straightforward. Sometimes that can be tricky, and it gets a lot of time to have a really good sequence, and of course as a cost in terms of time and in terms of money just for the sequencing. Second, analyze the mitogenome of unicellular organism is relatively easy because mitochondrial genome is abundant with respect to nuclear one, and it could be less complex to analyze in respect to the nuclear one, and also less complex than in metazoan, for example. The mitogenome structure itself and content of genes can allow further comparison. I mean, I'm just meaning the position of the different genes in the whole mitogenome structure. Then that can be another point for comparison with other closely related organisms. So just an additional tool to investigate evolution and phylogenetic relationships between organisms and different species. As a second very important point in our idea of renovating, let's say enriching the taxonomy, we look around for new concepts. And of course, what comes to our mind and just screening the literature is that the microbiome, hologenome, and the symbiosis, let's say, topics became really a hot topic in recent years. So we were thinking about the holobionts concept that just starting from a definition of limb margulis is the assemblage of two or more organism members of different species, which remain associated throughout a significant portion of the life history. What do we mean with holobiont? The holobiont is indeed as a single functional unit with its own characteristic that is different and new with respect to those of the specific parts. What I mean is, if we take the different bionts, the holobionts is not only the sum of these parts, but taken them together, they really form something new and unique. Some facts about the holobiont is that interested in this topic has recently awakened with new interpretations of the concept and theories on the underlying evolutionary driving force, like the hologenome, for example. And again, holobionts have been described for very many far-related groups of living beings, such as plants, algae, insects, corals, and even humans, but there are many more. And as last important fact that it's really important for our view is that stably associated organisms can influence their host physiology, adaptability, but mostly important also the morphology of the host. And as you know, the morphology is the fundamental parameter of traditional taxonomy. So for us, the holobiont concept would have been really an interesting topic to deep in our purpose. So in the end, for our idea of modern taxonomy, we thought it would be really useful to add from one side the concept of holobiont in practical terms, just insert stably associated organism as an additional descriptions, additional descriptors in our let's say characterization of organism, and of course also use the new tools that the next sequencing era, the next generation method in sequencing can offer us. So adding the host mitogenome to the description of the organism, but also the symbiont genome whenever present. So just to summarize, our proposal is to integrate the already integrated integrative taxonomy with more descriptors, namely the mitogenome and the symbiont genome and the holobiont concept. And in this way, create what we call the next generation taxonomy. And for test this new workflow, this new pipeline, we selected a model organism that our lab is really used to manipulate. I mean, a new species of Euplotes genus. Euplotes is really well spread and widespread in the environment, so it's easy to catch and it's easy to maintain in lab. And in our lab has really a lot of expertise in dealing with this kind of organisms. So we thought, okay, just try this new workflow on this kind of organism, and we tried it. So just going a little bit deeper in our workflow, of course, start with the isolation and culturing of the organism, and then go on with the so-called integrative taxonomy. So traditionally morphology method like live observation, and staining like Fulgen staining for the nucleus, silver staining for the ciliary pattern. Of course, also screening of symbionts and use ultrastructural method with SEM and TEM, and of course the use of mostly commonly used markers like 18S DNA and cytochrome oxidase and 16S DNA for the symbiont. But from the other side, we also use the more innovative techniques. So from a single cell, we perform the so-called whole genome amplification, and then we go on with the next generation sequencing through Illumina sequencing, and then we perform bioinformatics analysis to analyze the mitogenome and symbionts complete genome of the host when of the symbionts when present. We validate the information about possibly present symbionts with prop design and validative fish, and we want in this way to obtain a comprehensive holobiont description. So with this method, actually, we described a new species of Euplotes, namely Euplotes valluvenocchi, and to our descriptors that just like say in a traditional way, you have to provide to the journal to really give the complete description of the organism. We also added two new descriptors, as I said before. So the mitochondrial genome sequencing and annotation, and the description of the microbial consortium of this new Euplotes. So just to start, just to describe our achievement, we of course gave the systematic classification, nothing strange, is an Euplotes, so belonging to phylum Ciliophora, class Pyrotrichea, and so on and so forth. Then, of course, we provide the name, that is Euplotes valluvenocchi, and the etymology. We dedicated it to Anthony van Leeuwenhoek, the father of the microbiology, and for us was a kind of really important thing to dedicate this species to him. Then we provide, of course, the description of type locality, and guess where? It was in India. So for me, India has a really particular importance also in this study. So my colleague, my dear colleague, Dr. Nitla, collected the species in Kolleru Lake in Andhra Pradesh. And of course, we provide the type material through two different museums, the Calci Museum in Pisa and Natural History Museum of London. And I want to thank Alan Warren also for his help. And we deposit also some paratype slide also in the Department of Biology. I want to stress the fact that deposit type material is really important for this kind of study and the description of organisms, because in all the descriptions, often this kind of material lack, and it's really a pity, and it's really important to have type material to refer to in case of further studies in future. Of course, we perform the morphological description with very classical techniques, observation in vivo, Fulgen staining for the macronucleus and micronucleus, silver staining for the ciliary pattern. Of course, we also perform line drawing and SEM observation, and also the morphometry as a traditional description wants. Just to summarize our results, our species, it was quite tiny, so 50 per 33 micrometer in length and width. It has a C-shaped macronucleus and one roundish macronucleus. The Dargyrome was a really classical one, the double Aristomus type. It has 7-8 dorsal ridge with 13-14 decinates each, and present 3 furrows on the dorsal side. It has 22-29 hydraulic membranelles, and the series pattern was 10 frontoventral cirri, 5 transversal cirri, 2 marginal cirri, and 2 caudal cirri. Quite classical distribution of cirri, let's say, for Euplotes. Then, of course, we performed analysis for the fine structure, and we found the normal characteristics of Euplotes, but also a very interesting thing, we observed roundish particles in the cytoplasm, and these, please keep it for later. We, of course, perform gene sequences. The 18S was sequenced and deposited in GenBank. The best blast hit was with Euplotes antarcticus. And based on these results, of course, we also performed the phylogeny of our species that fall nearby Euplotes trisulcatus and Euplotes antarcticus in the clade A of the genus. Going on, we of course perform comparison with congeners, and we use the most close species from a morphological and phylogenetic point of view. And basically, the closest was Trisulcatus, and you may see that the overall body shape is anyhow different, and also in any case, molecular data confirmed that our species was new. And now coming to the real new descriptors, we were really proud to say that we succeed to get the whole genome, the whole mitochondrial genome, thanks to my colleague, Dr. Gamuto, who annotated the whole mitochondrial genome. We obtained a singular linear contig of around 41,000 base pair with 36 protein coding genes and 16 tRNAs. We also performed one analysis about the synteny of these genes, so the way in which these genes were distributed along the linear contig, comparing it with the Euplotes minuta and another Spirotrichea that was Oxytricha trifalax. And mainly we observed that the synteny was more or less always conserved. So this was another point in which the mitochondrial genome can be really useful and used to further comparison with the closest relatives. So last but not least, we observed and we studied the microbial consortium associated to our species, and from preliminary fish analysis, we saw that there were some bacteria inside the cell. So we went on, and based on our molecular data, we designed a species-specific probe for this new bacterium that we called the Candidatus pinguicocus supinus, and we detected it in a very peculiar part of the host cell, mainly underneath the cortex, and often distributed in two poles of the cell. From TEM analysis, we saw that it was a roundish bacterium with the diameter of about 1.3, 2.3 micron. And very peculiar, it presents really membrane invaginations or evagination that it's somehow peculiar of the group to which it belongs, and then we will briefly talk about. Often it was detected close to liposomes. So we performed the 16-layer DNA-based phylogeny, and we realized that our species fell into the phylum Verrucomicrobia in the class Opitute in the family Punisecoccaceae, with a rather long branch, probably due to a higher evolutionary rate of these endosymbionts. We also perform the phylogenomics based on 121 genes that confirms, in any case, the results of our previous phylogeny. And of course, thanks again to my colleague, Leandro Gamuto, we were able to sequence the whole genome of the symbionts, and we got that it has, we could obtain 168 predicted ORFs, 34 tRNAs, and 1 RNA operon. But what was really interesting to see, it was really small. This genome was really small, around 163,000 base pair. That makes it the second smallest known genome of symbionts in unicellular organisms, and the fifth smallest known genome in the whole literature about bacteria. Among the 168 ORF, we identified 131 COGs subdivided in this way. So mainly the most part was of the category J, so translation, ribosomal structure, and biogenesis. Then we have lipid transport and metabolism, and then post-translation modification protein, turnover, and chaperones. And another category where represented was cell wall, membrane envelope, and biogenesis, and envelope biogenesis. One really curious and striking thing is that the overall coding percentage was about 92%, and nevertheless, it was impossible to find genes related to cell division and genes related to DNA replication machinery. It means, and we also performed blast analysis on the host genome, but we did not retrieve any of these genes. So it means that these genes were not integrated in the host genome, and it means that maybe for this loss, severe loss of genes, our symbionts really live in close association with the host. It is our interpretation. So we also added the microbial consortium to our description, and we, in this way, succeed in perform a really comprehensive analysis of our host, of our ciliate. And we were really proud of that. And based on these results, we started to write down the project. So if you are interested in deepen these results, you may find our preprint on bArchive and the through publication on scientific reports that I reported here. So now, just to summarize the pros and cons of our method, of our workflow, that doesn't want to be the resolution of every problem, all the problems in taxonomy and systematic fields, but wants just to give a small contribution in the advancing of taxonomy in modern times. So for sure, one process that provides highly integrated in-depth characterizations of organisms, and we demonstrated it with the characterization of Euplotes van Leuwenhoekje. But from the other side, it requires a high level of integration among different biological disciplines, and it means a really long list of co-authors at the end of the work, that we are aware of that. Of course, ensures a rejuvenation of present taxonomy, because the present generation of scientists is really much interested also in deepen and explore the new techniques that genomics and bioinformatics gave us. Of course, for this reason, at present, can be more expensive than a simple traditional taxonomic study. We are aware of this, but we hope that in future things will change and this kind of analysis will become cheaper and cheaper. We think that is the right way, or just a good beginning, and we have also further future perspective. We would like to apply also functional studies and apply this workflow to higher level organisms, I mean small metazoans, and indeed we are just doing this in our lab with pretty good results, I will say. So the take-home message is for sure that the NGTax wants to introduce and try to introduce some novelties inside the present taxonomy, and in the other way, at the same time, it wants to keep alive traditional taxonomy and taxonomists, because we need taxonomists. We need them a lot, because without taxonomists, we can really give the real name to things, to our organisms. We need their experience, and we need to maintain this experience alive in the now and in the future. So we want to create collaboration and multidisciplinarity that can really last long. And of course, we demonstrate that this kind of workflow is suitable for in-depth characterization of unicellular organisms, and we hope that in future could be also used for pluricellular ones. I want to really thank all the co-authors of of this work on Neoprotis valluvenoffi, and of course also all my lab, or the lab from members from which I belong, and of course the organizers of the symposium. And really thank you to have invited me, and just thank you for your kind attention.
Now, I invite questions from the audience, please. Any questions from the audience?
Yes.
Any questions? If there are no questions, now I take the opportunity to thank you, Dr. Sarah. Thank you for your enlightenment. And now over to you, Rimpi. Thank you, Dr. Sarah. Over to you, Rimpi. Thank you.
For conducting the session. Moving on, I would request the co-chairs of the next session, Dr. Ilmas Naqvi, Associate Professor, Department of Zoology, Zakir Hussain College, and Dr. Tajinder Kumar, Assistant Professor, Department of Zoology, Mathri College, University of Delhi, to take over and introduce the chairs for the session.
Good afternoon, one and all present. I'm the co-chair of this session with Dr. Tajinder. First of all, I would like to express my gratitude to the organizers for giving me this chance. I will now introduce Dr. Geetu Gambhir, one of our session chairs. Dr. Geetu Gambhir is professor at Department of Chemistry, Acharya Narendra College, University of Delhi. She is an organic chemist with doctoral degree in synthetic chemistry for biologically active natural chromomes and coumarins from Department of Chemistry, University of Delhi. She has been long engaged in the research and development of ion selective polymeric sensors, biopolymers in the past completed projects. She also holds expertise in synthesis of nanocomposites and further tailoring them to adapt to the acquired need of optimized sensing under the defined conditions. She has successfully completed 3 projects and have number of publications to her credit. She has authored 2 books and 4 e-chapters. She has been a resource person at Institute of Lifelong Learning. She has developed number of modules for educational content in e-domain. She has been on the judging panel of Inspire and other prestigious schools of Delhi. She has mentored more than 30 students for projects at undergraduate level. Moving on, now I would like to request Dr. Tajinder to introduce Dr. Renu Gupta, our other chairperson. Over to you, Dr. Tajinder.
Thanks, Dr. Naqvi. I'm happy to introduce our chairperson of this session, Professor Renu Gupta. Professor Renu Gupta is a professor of zoology at Mathri College, University of Delhi. She did her MPhil and PhD in zoology from University of Delhi and has been awarded postdoctoral research associate fellowship of CSIR DST Women Scientist, North-South Dialogue Scholarship, and Ernst Mac Grant Fellowship. She has authored a book and 3 book chapters. She has published many research papers in international repute. Her research has been applied immensely by the scientific community on various platforms. And she has received the Best Presentation Award, Innovative Research Award, and Women Scientist Award in the Applied Zoology. She has been teaching ecology, cell biology, molecular biology since 2001. She is also a mentor for Students Project at Maitrey College since 2010. She has also been a reviewer for reviewing research manuscripts submitted for publications to Zoological International Journal since 2009. Welcome, Professor Gupta. Over to you.
Thank you, co-chairs, Dr. Ilmas and Dr. Tejinder. It is an honor to introduce the speaker of this session, Professor Sergei Fokin. Professor Sergei Fokin is a professor at Department of Biology, University of Pisa. He earned his PhD from Leningrad State University, Russia in 1978. His scientific interests include protistology, functional morphology of ciliates, biodiversity of the protists and their symbionts, symbiotic relationships between protists and bacteria, and history of zoology. He has been an inviting professor of Yamaguchi University, Japan, and Pisa University. He is also a leading research fellow of St. Petersburg State University, Russia. He has collaborated with various universities and institutes from Germany, France, Poland, USA, Canada, and Japan. He has authored more than 450 publications and has many awards to his credit. To name a few, a medal and prize in biology for young scientists by Academy of Science of the USSR, a medal in ecology by Lomonosov's Foundation, Russia, Collis Award for Ciliate Systematics, USA, a diploma and prize by St. Petersburg Naturalist Society for History of Biology, Russia, and the best protist image at the 15th International Congress of Protistology, Prague. The title of his today's talk is Ciliates and Their Bacterial Symbionts, Ecological Point of View. I request and welcome you to deliver your talk. Over to you, Professor Fokin.
Dear colleagues, are you hear me?
Yes, sir, you're audible.
Thank you for the nice introduction. I'm going to switch on my presentation. Do you have a look on it? Can you hear
Yes,
me?
Yes,
Okay,
sir.
so dear colleagues, I'm really happy to join this meeting, especially because almost 15 years ago, I attended the first such a meeting in Delhi, and I have very nice memory about that time in India. Today, as you can see from the first slide, I am going to make quite short overview of the topic, which nowadays very much rapidly developing. And then, of course, each year we can find something novelties in this area. So talking about ciliates, I should go back to this rather well-known for everybody picture, came from the publication of UK colleagues, and they are present biodiversity of Ciliophora, even according to position, which was published almost 3 decades ago. We are convinced that it should be just 8 or 9 1,000 species. In reality, I believe calculation should be enlarged at least twice or even more, because aside of the such a giant, as you can see here, like Stentor or Spirostomum or some colonial ciliate protists, we have a number of very tiny organisms like Cyclidium, which has a dimension around of 10 microns only. And that means that a number of biodiversity is still out of our focus because they are so small. They are partly commensalists or symbionts in other organisms, sometimes protists, sometimes metazoans. And definitely we should keep in mind that true biodiversity of Coleophora much bigger than we could expect according to this calculation made by UK colleague. So today we are going to discuss, of course, very briefly, particularly sub-lineage Coleophora, which is a part of Superphilium alveolata, with two sister group, Dinoflagellate and Apicomplexa. Of course, they are unified just because, according to the phylogenetical reconstructions, they are monophyletic sublineages. But of course, our main point is Coleophora. Nowadays, we have a number of tools for investigate not only morphology, but also even evolutionary history of this group. And according to some of such a reconstruction, we could find that origin of ciliated protists can be lasted back almost close to 2.2 billion years ago. So roughly speaking, they originated in the same time as a eukaryotic cell. So they are very ancient, and then they have a lot of time for evolution, for splitting some particular sublineages, as you can see from this slide. Mainly, we are talking about kind of model object. It's common procedure for ciliatology. And first of all, of course, we should select for such a point of view Paramecium as a well-known, well-studied, but still under investigation genus of Oligohymenophora, but also several other groups could be treated. In fact, particularly I was involved in such a business of investigation, Paramecium and Frantonia sister group for Paramecium for a very long time, and would say that I am specialist about this point.
Okay. Are you moving the slides because we are still on your title slide?
Sorry?
Are you changing the slides because your slides over here is only on your title slide? Yeah, now I can see them moving. Can you just shift to slideshow?
So now it's moving?
Yeah, now it's moving. But can you shift on slideshow so that it's convenient for you?
Yeah, okay. So talking about this point, we addressed our attention for nowadays classical phylogenetical reconstruction of Coleophora. So in many textbooks or in many reviews, you can find such a splitting according to the molecular markers, all biodiversity inside of 11 classes. As you can see from this slide, some of them marked because in one of them we can find quite many differently looking and differently communicated with the host cell type of symbionts. So in reality, nowadays, phylogenetical reconstruction of Coleophora going to improve or let's say become more complicated because at least 6 new classes were added recently for such a phylogenetical reconstruction. And it's again indication that even such a classical systematic or taxonomical field of investigation, very much important and very rapidly developed in the last several decades. So among of that new classes, we can find several of representatives which really very much interesting for us because, for example, Parablepharismia, one of the sublineages totally populated by ectobacterial symbionts. In case of Copemetapea, which we just going to describe as a new classes in our publication, which in press now, we also can find very peculiar type of symbionts and so on, so forth. So just to illustrate it, what kind of model objects we are going to discuss partly in my talk. It is a Paramecium genus. Here you can see some of most well-described and highly accepted morphospecies of Paramecium genus. Meantime, several of them were redescribed or described by our team. So you can see this quite old lineage in Oligohemianophorea classes, namely subclasses Piniculea, rather diverse according to the morphology. Let's say they have a rich morphology, and then we can discriminate them quite easily according to the morphological characteristics. But of course, we're also nowadays using a number of molecular tools. The same could be addressed to a sister group of Paramecium Frontonia, which may be even more under investigation last decade. And again, this is a great community right now, more than 30 species. In one of them, again, we can discriminate it quite differently looking type of ciliated protist, which moreover populated all possible ecological niches. So we can find them in the marine environment, in brackish water, in fresh water, or even in soil. So some of my examples will be connected with this group of ciliated protist. So finally, according to systematic current situation connected with the Paramecium and Frontonia, we can address our attention to this phylogenetical tree, which easily can show you that they are much diverse according to the phylogenetical reconstruction point of view. So the Paramecium could be split nowadays in 6 different subgenus. Among of them, for example, Gigaparamecium, which quite recently was included in a Paramecium genus. It former Neobursaridium gigas, but according to recent publication, it is a giant Paramecium. Okay, the same could be addressed to Frontonia, which nowadays splitting at least for 3 sublineages, which meantime could be treated as a different genera, and such a review process still in front of us. Okay, so what's the main message which I would like to give you during this talk? Bacterial endosymbionts or symbionts or endocytobionts are largely undiscovered world. So a number of knowledge even we can complete it each year, still not far from a real picture of biodiversity and way of communication between protist, Paramecium or Frontonia or some other, and such invaders as a prokaryotic organism bacteria. For that point, we should address our attention back to this very well-known type of definition made for symbiosis by Anton de Bary. We already saw this person as a portrait, of course, during the early presentation. And he made very short but precise definition what symbiosis is. Close association of individuals of different species. It means such a definition covered quite precisely all possible way of communication between host organism and invader. So it could be mutualistic symbionts, like we can find here for crab and sea anemones, a number of representatives which manifested obligate symbiosis between different organisms like Radiolaria and former so-called yellow cells, which in fact dinoflagellates. Again, example of true symbiotic relationship. And meantime, a number of parasitic system in which one living beings which are parasitic populated different structure of another organism, namely ciliated protist, like you can see here, situation of microsporidia inside of Euplotus cytoplasm, or Stichotrichia representatives, macronucleus of which populated by Leptomonas-like parasite. For that point, I would like to remind you several definitions which totally address to parasitic organisms. But again, keep in mind that parasites are kind of symbiotic relationship, even a bit different from relationship between partner in that system. So as you can see from this slide, in majority of cases now parasite treated as a invader which able to partly or completely donated a number of function to host organism. Mainly it connected with a relationship, and that such a concept could be treated as an ecological one. Thus, features of topology connected with a bacterial symbiont and their relationship with the host cell can be considered also as an ecological. And therefore, ecological aspect has an intracellular and extracellular level of consideration. Talking about subdivision of possible and the symbionts or symbionts, usually we can discriminate it three major groups. Accidental invaders or invasion, which could be a kind of mixed community, but some of them also could be a parasite. Permanent and the symbiosis relationship, which means mutualistic symbiosis, and highly infection bacteria, which, as I show you in a few minutes, should be treated as a true parasite. So talking about topology of such invaders, easily we can discriminate ectosymbionts and endosymbionts, or let's say ecto and endobionts, because again, way of communication between such an invader and host organism could be different and not always very easy to discriminate what kind of definition fits mostly for particular case. So you can see here some of ecto type of symbiosis between bacteria, for example, in Verticella, or in metopus, or kind of intracellular invaders. Again, we can find some of them in metopus, representatives of Armaphorea class, or inside of the different sublineages of Oligochymenophorea, like Verticella, which could populated by microsporidia. The same could be addressed to Frontonia, or even in some cases, one ciliated protist could be populated by autotrophic type of symbionts, mainly representatives from Chlorella group of green algae. So talking about this ectosymbiotic type of relationship, we should remind ourselves several representatives. First of all, a very famous case of Kentrophorus, which actually should be treated as a system of necessity type of invader, such a bacteria which nowadays treated as a gamma proteobacteria ectosymbionts, Candidatus kentros, populated dorsal side of the host organism, and they are really necessary because this type of ciliated protist has no oral aperture. It could accept the food only via back side of the body, and such a vegetable garden can be eaten by host organisms. So it strictly connected with such a relationship between host organism and bacteria. Almost the same, even it not yet so well investigated from physiological or let's say biological point of view. It is a case of epibiont belong to theotrophic type of bacteria, which totally populated surface of Zootamnium niveum. You can see a colonial of this Pyretrich, marine Pyretrich, which all zooids totally populated by bacteria. By the way, it's representatives of gamma proteobacteria group. So, and finally, we also can address our attention for another famous example of ectosymbiosis. It is ectosymbionts which covered by particularly strip of dorsal side in a Euplatidium itoi. They are verrucomicrobia, quite a rare case. Even in the previous talk, you already saw that it could be possible to find symbionts from that group. But nowadays, such a bacteria also under consideration as a possible model for searching origin of flagellum or cilium. So that quite important example also from evolutionary point of view. So in reality, as you can see from the next slide, we can find a number of such ciliated protists which are totally populated by ectosymbionts. Definitely they are necessary for the host organism, especially because majority of these ciliated protists are a member of anoxic or little oxic community. So they should use another way of energy transportation, and quite probably majority of these symbionts could participate in this chain activity. So parablepharisma, as you remember nowadays, it's separate classes. One of such example, you can see how many bacteria, ectobacteria could be. It's a sulfur bacteria. They moreover communicated with the cortex of host organism in a very peculiar way, and they always closely located from outside with hydrogenosomes, because again, they are anoxic or little oxic member, I mean, from ecological point of view. And at that situation, classical mitochondria doesn't work, and they are replaced by hydrogenosomes. So, as already was stressed, some of quite close relative species of ciliated protists anyhow could be populated by rather different bacteria. Another representatives also belong to parablepharisma. They are not yet described, but again, a surface of cortex populated by very distinctively looking curved or straight bacteria, which probably should be treated as a part of the life cycle for the same organism. But what is important in that case, as you can see from this slide, they even manifested very peculiar ultrastructure, which remind a bit plastid or some other machinery connected with photosynthetic activity. They produce sulfur particles, and probably this is a basement for such a synthesis. Actually, such a structure indicated for bacteria for the first time altogether. Moreover, another group of ciliated protists like Plagiopila, sublineages Plagiopila, its particularly classes, and the majority of them also a member of anoxic community. One of them, Sanderia vorax, you can see in this slide, and you can recognize how many bacteria very precisely distributed between the longitudinal road of cilia. So the dark line are cilia road, and after fish reaction, all bacteria visualized as a red particle. Again, talking about this system, we can recognize that it's a lot of bacteria. They are distributed quite randomly on the surface of body, but they are not communicated in a host organism in the same way as you saw previously. So they're embedding in a mucus layer and mainly distributed horizontally according to the surface of cortex. So definitely way of communication in that case could be a different. Okay, in majority of cases, if we are shifting our attention from outside to inner structure of ciliated protist, we can recognize a number of bacteria which somehow adapted to live either as a true symbiotic, mutualistic symbiotic invader, or as a parasite inside of the different subcompartment of ciliated protist. In fact, it means that majority of potential invader should arrive using food uptaking process, phagocytosis. That's very well described type of machinery in a ciliated protist, and you can see that in different way escape from the food vacuole. Such an invader can be accommodated in a nuclear apparatus, even between the membrane of nuclear envelope, or in Golgi apparatus, or sometimes in the mitochondria, or in a particularly Golgi, particularly in a cortex layer. So the main strategy of any invader, doesn't matter which they are, should be just to find the right way which prevent contact between invader and host lysosome. So they should be kept in a new location, and then they should be distributed in a progeny. So these two major tasks for any invader, also for, generally speaking, all type of parasite. Anyhow, it's probably not a unique way of entrance to the potential host organism. Quite a long time ago, it was description in a Parauranema acutum. It was coined as a xenosomes. And as you can see from this slide, which is an extraction from Solda and co-authors' articles, they able to rupture it cortex directly. So it's kind of lysis process in which invader prokaryotic organism able to enter in firstly to alveolar vesicles and then directed to cytoplasm. It's very important to remember such a case according to the evolution, because you know, symbiogenesis, especially if we are talking about origin of eukaryotic nucleus, always indicated possibility to communicate between two different prokaryotic organisms. How they could communicate it without phagocytosis? Probably that good example, they able to use a lysis process to invade one prokaryotic cell by another. Anyhow, talking about general composition, we easily can recognize that Paramecium should be treated as a true model object, composition of different type of invader, different type of symbionts, almost the same as we can collect in a Coleophora, generally speaking. So just to have a look on several representatives of such a cytoplasmic symbiont, we should of course mention this bacteria symbiont of Euplotus community. It already was mentioned that Polynucleobacter or Polynucleobacter-like bacteria belong to the beta Proteobacteria group, quite randomly populated cytoplasm of many freshwater or brackish water species belong to Euplotus. As you can see from this slide, even morphologically, far not of them identical with the Polynucleobacter. Here you can see the classical ultrastructure of Polynucleobacter necessarium. Moreover, that group of symbionts also can affect somehow quite fundamental process of replaced cortex structure. So morphogenesis also can be connected with the present or absent of symbiotic bacteria. If in previous case, Polynucleobacter definitely can be affected process of food uptaking or digestion process, if of course we remove such a bacteria from cytoplasm. We can find another result if we remove such a cytoplasmic bacteria from Euplotus daedaleus. You can see they produce a number of abnormalities in a composition of ventral side, which means that they are somehow necessary for classical type of communication between host cell and such a symbiont in a wide sense according to the Debarry. Finally, we can recognize inside of the cytoplasm subcompartment a lot of different ways to communicate between bacteria and host structures. So sometimes they even manifested flagella. They could be necked or populated by some symbiontophoric vacuole, or sometimes lying necked without any decoration, which means that they have somehow defensed against fuse with a lysosome, but with another way. Or they simply can be decorated by endoplasmic reticulum vesicles came from the host organism. Again, with the same idea, they should be in a safety place according to the communication with a possible invader. So talking about such a way of communication between host organism and symbionts, definitely we should address our attention for multiply symbiosis, because quite often either in a cytoplasm or in a nucleus, we can recognize simultaneously several representatives, quite often not so closely related to each other. For example, here, one big particle belongs to the infection form of Halospora obtusa, Halospora clade, instead of a small one, which also manifested the same construction of the life cycle. It's Priria caryophylla, still Halosporacea family, but not the same. For that point, we should switch our attention for case of intranuclear type of symbiosis. So such a Halospora-like bacteria, exclusively cellular type of symbionts of Paramecium species. You can see some old picture extracted from literature from the middle of 19th century, and original picture illustrated different type of such a bacteria. What is very much important for this point, they manifested very special type of the life cycle, splitting in an infection form and reproductive form. And as you can see now, this community is rather huge, and among of the classical Halospora bacteria, we also can find Halospora-like bacteria, which manifested the same strategy. So they have reproductive and infection form, and they are able to infestate experimentally asymbiotic strain of host organism in a laboratory. Another point which very much intriguing that such a bacteria, especially Halospora-like bacteria, could be detected not only in a Paramecium, but in many other representatives of ciliated protists. So here you can see several fish preparation dedicated exclusively symbionts from Paramecium species like Curviuscula or Obtusa, or again Obtusa, different step of the life cycle. So classical Paramecium or Paramecium Halospora like bacteria like Gertsia or Priria. As I told you, we can find such an infection also with some other group of ciliated protists. For example, here they are in a micronucleus of Frantonia, but also we can detect it in a Prorodon, completely different classes, or even in a Helidonella, third classes of ciliated protists. One of major peculiarities which help us to discriminate these two groups, namely classical Halospora and Halospora-like bacteria, it is a way how they can modify division process of the host organism, because they're able to use host machinery for discriminated infection form, which then after concentration in the middle figure of division, releasing to environment and sources as a infection form for the distribution of these bacteria in a population of hosts. Instead of that, Halospora-like bacteria lost this machinery, or maybe it's still not developed this machinery. So probably this group of holospora-like bacteria, from evolutionary point of view, should be treated as a more ancient. So especially important that such a invader, which again indicated that they are rather parasite, because they manifested all peculiarities of parasite well known from relationship between such an invader and a number of metazoans. So they able to change machinery of host organism using genetical reconstruction via sexual process, conjugation. So if we have a look on fate of such an infestated macronucleus by different bacteria, we always can recognize that they able to shift normal reconstruction of nuclear apparatus in such a way that finally next generation of ex-conjugant always could be infestated by bacteria. So finally, they either able to keep old macronucleus, which usually degraded during the conjugation, or it can produce chimeric macronucleus, partly consisted of new unlagged, but partly populated by bacteria from old macronucleus fragments. Okay, so coming back to the main topic, we can stress that ecological significance of symbiosis between ciliated protist and bacteria in a natural community, not so much investigated up to now, and we still have quite a little about real situation and way of maintenance of bacterial infection in a ciliates in a native population. Another point which we should briefly touch, why majority of bacterial symbionts of ciliates only are facultative and not obligate one if they could be true symbionts. My answer is that because they are parasites, and definitely they communicate with a ciliated host organism as a true parasitic invader. Then host organism definitely should create a kind of defense mechanism again of such a infection. And we can find it as a bacterial lysis quite well spread either for micronucleus or macronucleus, which can be populated by bacteria, but bacteria very fastly lysed inside of the karyoplasm. Quite unusual situation for basical example for cell biology. Another very important from biological or even evolutionary point of view example that such a infection bacteria like holospora able to use infection form of own life cycle as a vector for introduction, not infection form of bacteria inside of the nuclear apparatus. You can recognize that inside of the food vacuole, always they collected a lot of different bacteria, and when infection form escape from the food vacuole, they simply can take with them some of non-infection form. So such a co-infection free-living type of bacteria or simply not infected type of invader with the holospora experimentally was proved. You can see such a scheme, how such a novel symbionts can arrive to the final destination, to the macronucleus, and then quite often overgrowing the vector, the holospora infection form. So this is a way how we can discuss biodiversity of symbionts. So it's a huge area of investigation, as you can see for this slide, dedicated to phylogeny of Holosparaceae. Nowadays, we can recognize several clades of such an infection type of bacteria: classical Holospora, Hafkinia, Gyortzia, Priria, or even some still not well investigated or single type of invader like Paraholospora nucleovisitans, which able to create infection both in cytoplasm and in a maker. You see, this is a lot of possibility to investigate this phenomenon in a laboratory condition. Finally, I would like to show you some reconstruction, which partly could be treated as an evolutionary sequence, partly just as an illustration for a major way of adaptation, such an invader, parasite, or in future symbionts in a host subcompartment. So majority of them arrived via phagocytosis. In cytoplasm, they able to escape from the food vehicle or decorated by themselves or with the participants of host machinery by membrane, which predict fusion with a lysosome, or they can be transported in a particular way to other subcompartment like nuclear apparatus or cortex or even mitochondria, into which they basically defense against communication with the lysosome. Simultaneously, it's very good location for be distributed between progeny. So they solved all major problem of classical parasite. From evolutionary point of view, especially important that, as you can see, they able to be introduced in inside of the mitochondria or plastid. It means that one basically prokaryotic organism can communicate with another prokaryotic organism. Again, this is an illustration how we can discuss possibility to create an eukaryotic cell, especially eukaryotic nucleus, using two different type of prokaryotic organism, eukaryotic, bacteria and archaea. So finally, just coming back to kind of ecological point of view, we should have a look on such a map of distribution bacteria, which classically treated as a Holospora or Holospora-like bacteria. And it is very easily to recognize that such a map mainly connected with the distribution of researchers. So where we have a number of people involved in this business, we have a number of results. In many other places where we do not have either professional protistologists or simply such a territory never been checked by professional protistologists, we still don't know what's going on. For a long time, it was coined an idea that such a symbiont mainly depend on the climate and mainly distributed in a temperate or even cold climate area. But as you can see, step by step, we can recognize some of them in a subtropical or even tropical places. It means that it's a really huge open field for the further investigation, and definitely we will do it in the nearest future. Okay, so this is a quite short overview which I would like to present for you, and thank you for your attention. That's the end of my talk.
Thank you, Professor. If anybody wants to ask any question, then most welcome. We can only take 1 to 2 questions. Okay, so we are thankful to you, Professor Sergey Fokin, for an enriching presentation, and it was full of the knowledge about ciliates and their biological symbionts with the new dimensions towards the ecological point of view, such as morphospecies of the paramecium and frontonia are re-described on the basis of the mitochondrial genes, ecto and endobacteria of various species and their intercellular localization mechanism, electron micrographs, behavior of symbionts during sexual process of the host, all were really fascinating. So thanks again for enhancing our knowledge with your years of your expertise, and we would be more than happy to be connected with you through email, and also would be like to see you again in our upcoming more events. Thanks a lot, Professor Sergey Fokin.
Thank you so much. My pleasure.
Thank you, sir, for summarizing the talk and concluding the session. We will now proceed to the next session, for which I would request the co-chairs, Dr. Shalu Sachdev, Associate Professor, Department of Zoology, and Dr. Rimpi Chauhan, Assistant Professor, Department of Biomedical Sciences, Acharya Narendra College, University of Delhi, to introduce our learned chairs to the session, to the audience.
Thank you so much. It's my pleasure to introduce to our chairpersons for the session. Dr. Geetika Kalra is an associate professor at Acharya Narendra College since 2005. She has obtained her BSc, MSc, MPhil and PhD from University of Delhi. She specializes in physiology of adventitious root formation and has 10 research papers to her credit in national and international journals, and has authored approximately about 5 chapters in a book on plant physiology. She has also contributed to developing e-content for the university. At her institute, she has been convener for annual cultural committee and organized various gala events in the college. Over to Dr. Shalu.
Shalu ma'am, please unmute.
I'm so sorry. Good evening. It gives me an immense pleasure to introduce the chair for our session, Dr. Anita Narang. Dr. Anita Narang is an associate professor in the Department of Botany at Acharya Narendra College. She obtained her MPhil and PhD from University of Delhi with specialization in plant tissue culture. During her post-graduation, she specialized in cytogenetics. Presently, she is working on mushroom cultivation in the college. She is the convener of Shashak, the Women Empowerment Society of our college, coordinator of biological domain of VLab, and she is a member of 3 other societies. Besides this, she has been actively involved in many extracurricular activities of the college. She has 5 publications in international and national journals, along with 3 chapters in books to her credit. Over to the chair.
Thank you, Shalu, for your kind words. It's a privilege to introduce Professor Bettina Sontag. Professor Bettina Sontag obtained her diploma and her PhD from University of Innsbruck, Austria. Her doctorate work involved study on seasonal dynamics of ciliates and flagellates in a deep oligotrophic lake. Presently, she is the head of research group on ciliate ecology and taxonomy at the University of Innsbruck. Her research group focuses on ecology, biodiversity, taxonomy, and phylogeny of ciliates, mainly in the lakes. Professor Bettina has about 50 publications in national and international journals and 5 projects to her credit. I would now like to invite Professor Bettina to deliver a talk. Welcome, ma'am.
Thank you very much for the nice introduction. I will now start my presentation. Can you see my presentation and hear me?
No, ma'am, not yet.
Okay. So. Can you now see and hear me?
Yes, yes, ma'am.
Okay, thank you very much. Yeah, thank you very much for the nice invitation. And it's my pleasure to talk about planktonic freshwater ciliates and about their key roles in aquatic environments. So when you take a look in a drop of water, you see many, many different protists and bacteria, photoautotrophic bacteria, cyanobacteria, or heterotrophic bacteria, also phytoplankton, heliozoans and amoeba, viruses, and of course, many different heterotrophic and mixotrophic flagellates, but of course also ciliates. And when you take a look onto these numbers, it's really incredible how many there are in just a drop of 1 milliliter of water. And of course, they are all interacting and communicating among each other. The ones, some are food for the others and some are predators on the others. And when you take a look through the microscope, I'm sure that many of you saw more or less this image. There are many, many of the microorganisms. And in my research group, we are particularly interested in the identification and the specific role of a ciliate species in its environment. So we pick a certain ciliate of desire and relate it to, for example, algae that are present in the environment and try to figure out which ones are food for the ciliate or which ones are predators for the ciliate. But why study ciliates in their environment? Because, as you heard also before, they are key organisms and also in all aquatic environments. They belong to the most important consumers of algal biomass, but also bacterial biomass in pelagic food webs. It depends always on the trophic status of an aquatic water body, which ciliates may be predominant. And they are an important link between lower and higher trophic levels, which means they feed on bacteria or phytoplankton, other protists, and they are in turn also fed by metazooplankton, for example. They are excellent indicator organisms because they have short generation times of hours and days, and they are able to adapt because of these short generation times to changing environmental conditions, and not even the species alone, but also the whole community changes. And of course, they are excellent model organisms, as we have seen before in the previous talks. And you can also address ecological and ecophysiological questions in lab experiments. So our aim in our research is that we find correlations between a ciliate species and other organisms that are present in the same water sample. Here you can see 4 examples, 2 from one lake, but from different depths. Here we have a sample from 5 meters depth and another one from 40 meters depth, and the same for another lake. And when we focus on one ciliate species, of course, we have different communities of microorganisms in the same water sample. So also the relationships between a ciliate and the other organisms change according to the environmental conditions you have there, or also to the organisms that are in the same samples. And so you can draw conclusions on the organisms which are food, which are predators, or which are even symbionts. The interpretation of these findings require knowledge, of course, on the nutrition mode of a ciliate, which means you need to know if it's a bacterivorous species or is it an algivorous species, because you also have to consider the identity of the food algae if this algae can indeed serve as a food item. For example, if you have a very large algae that cannot be ingested by the ciliate, then of course it's just a co-occurrence and it's not a predator-prey relationship. So you also need to know the identity of the potential food item and not only know that it's an algae. And of course you need to know if the ciliate, for example, has defense options, if they may possess some extrosomes so that a predator can be expelled, or that they have, for example, a jumping behavior so that they can easily escape predation pressure. So you need to know a little bit more on your ciliate and also on the other organisms that are present in the same sample. The ciliates are highly diverse, and if you have a look onto these images, then you can easily recognize that they are very different. They have different sizes, different shapes, different ways of life. And they have many different lifestyles. And this I would like to elucidate here, because we have here 4 ciliates that belong to the same ciliate group. We have 4 peritric ciliates, and we can see that we have the typical appearance. We have a cell that in most cases has a stalk, and with this stalk in the plankton, these peritric ciliates are attached to, for example, diatom colonies, or they can also be attached to zooplankton. They can be freely floating, and so on. So you see, there are, at least in the same group, there are differences how they move or just sit and are moved around. Then what else you can see here is that you have these transparent forms. These are heterotrophic ciliates. And from this heterotrophic nutrition mode, for example, in these ciliates, you can see that you could even identify the food items. So this is very important that you also have a look onto the food and which algae, for example, were fed by this ciliate. But there are also other ciliates, and again, they are in the same group of the peritric ciliates. I saw the others here. And they have a grass green appearance because they bear algal endosymbionts, or zoochlorella. And these algae independently live inside the ciliate cell. They divide there, and they are also given to the daughter cell after division. Then these ciliates can get photosynthetic products from their symbionts, but they also actively feed on food items, on other food particles. And this nutrition mode is called mixotrophy. But there is another way of mixotrophy, and this is shown here because this ciliate species, for example, is also feeding on algae and digests everything from the algae except for the chloroplast. And the chloroplasts you can see inside the ciliate. they are left over, and the ciliate still gets photosynthetic products from these stolen chloroplasts or kleptoplasts, how they are called. So you see, even if we have ciliates from the same group, they have very different lifestyles. And again, here I would like to elucidate that even from the same genus, we have heterotrophic and mixotrophic forms, and therefore it is important that the species is identified. And it's not in respect to an ecological investigation, it's not sufficient that you only determine a ciliate down to the genus level. And of course, what is also important here is that the green algal endosymbionts are identified, because in the most of the former studies, they were described as chlorella-like, or so chlorella, which is correct, but there were not the molecular tools available that we also have now. And so we are also aiming to identify not only the ciliates, but also the endosymbionts. And for example, for Coleb species, we found that they all had Micractinium conductrix as an algal symbiont. And for Paramecium rosaria, there already several endosymbionts have been found. What else is important when it comes to ecology is the symbiont specificity, and if a ciliate always bears the same algal endosymbiont, or if this symbiont can be exchanged by a different algal species. And last but not least, the nature of the symbiosis is, of course, also interesting to investigate if a ciliate can live also without the endosymbionts or not. So when we start with our ciliate investigations, we normally follow a traditional approach by microscopy, and we get available information on the species identity or also on the community composition of a water sample or in a water sample. Following the traditional way, we always use these, what we call the ciliate Bibles of Willi Voisner and a microscope. And I'm sure that many of you who work with ciliate identification know these blue books, but unfortunately 4 of them are in German, but there is also an English guide, at least to the ciliate groups, but also to the species. Yeah, and the plankton book is in English. And what is really remarkable here, and upon their release, everything, all the knowledge on a certain ciliate species, including also notes on their ecology, are included in these books. And what you can find there, as I said, you have the key to the species, to the groups, to the species, and you can also find a description of ciliate communities. And here, for example, you have a community with species that can be found in healthy activated sludge, for example. Here you have the community, the Cyrtophoridea, which are quite common in clean rivers, or you have the Oligotrichidea, which bear ciliates that are commonly found in freshwater lakes. So the habitat shapes these ciliate communities. And here, as an example, you have some planktonic species, and you have a lake, you have the lake surface, you have the lake bottom, and you have abiotic, but also biotic gradients, but it's just an example. But in the lake surface, for example, you have species that feed on algae because they have algae available in the light flooded areas of the lake. You have also mixotrophic forms in these upper layers of a lake. And when you go a little bit more downwards towards the lake bottom, you might have more bacteriovorous species because you might have less light or no light available. And you might also have species that are able to deal with less oxygenated and colder abiotic conditions. And you might also have anaerobic ciliates, and as Sergei already mentioned, they have the hydrogenosomes also to survive in these non-oxygenated areas. Then you can also figure out, for example, the ciliate communities that live especially in these upper water layers, and it is also important that you relate them, for example, to the solar radiation that can penetrate the water column. And there you can find very specific ciliate communities, especially also, again, these mixotrophic ciliates. Then there is another group of ciliates that is more surface associated. And these surface associated ciliates you find more in the shorelines and where you have more plants or stones and where they can crawl and attach to surfaces. So you have very different ciliates or areas, of course, in a lake where you have different communities that are shaped by the environment. When we study plankton diversity, we normally check out which ciliate species are there in the course of the year and in which depth also. So we can see ciliate communities, for example, in the colder colder months of the year, and they change considerably in spring and summer. And again, for example, we can then see these mixotrophic ciliate species that prevail mainly in autumn. And here is just a sketch again about the phototrophic assemblage containing more algivorous, mixotrophic or omnivorous species prevailing in the upper layers of a lake, and the heterotrophic assemblage that we can find deeper down in the lake. Then from our approach, we take water samples with a water sampler and do some quantitative protargol staining. This is a widely spread method when you do ciliate ecology, because you can at once identify the ciliate and also estimate their abundance. So we can count and see who is there in the lake and estimate the spatial and seasonal occurrence of a ciliate species throughout the year and along a depth gradient. And here, as an example, you can see we have a ciliate species, and the ciliate obviously has two peaks. Now we can, of course, relate the ciliate's appearance to abiotic parameters, but what else we can do is we compare it also to the appearance of other species. And in case of this ciliate, we found that there was another ciliate present that led to the extinction of this former or this ciliate species, because it was a parasitic Suctorian species that parasitized our host species, for which we also estimated the abundance. So when you take a look into the microscope, you can see that there were 2 ciliates present and that the one parasitized the other. And for example, if you would have only estimated the ciliate diversity from molecular sequences, then you would have missed this important relationship. So a look into the microscope is also worth to be done. So again, the traditional approach provides valuable information, of course, on the species identity, on the community composition, and also to figure out the relationships among ciliates and other microorganisms. Then you can also see their abundance and distribution along time and depth gradients in a water body and correlate the data with biotic and abiotic parameters. But even if you have figured out the connections among ciliates and the potential food or predators, you need to go to the lab and perform experimental approaches to confirm the findings. And here is an example of an experiment where we tested appropriate cultivation media, for example, for a certain ciliate, and also specific food items that we isolated from the same lake. So we have cyanobacteria and also algae that we fed the ciliate and figured out if they were an appropriate food item or not. But of course, for this, you need to isolate not only the food, but also the ciliate and be successful with both having them in the lab culture. Or another example is that you also test a ciliate against its sensitivity to solar radiation. You can cut off certain wavelengths. For example, here we cut off the UV radiation, and here we have the full radiation and also control. And so you can just check if this abiotic factor or this environmental factor was indeed influencing your ciliate. Yeah, but state-of-the-art approaches do not only include the traditional approaches, but also the molecular and bioinformatic tools, as also Valentina already impressively showed. So I would just like to give you some examples on what we did in the last months. Sorry, was there? So one of the examples is Coleps. And Coleps, maybe some of you know Coleps species. These are these nice ciliates with this armor, and they are very common in freshwater habitats, not only freshwater, also in brackish water you can find Coleps species. Can you hear me now again? Yeah. Okay. And more than 40 species are known. We again have heterotrophic and mixotrophic Coleps species, and commonly we find 3 morphotypes that were identified as Coleps hirtus hirtus, Coleps hirtus viridis, and Coleps betae. And because of a close look into the microscope, I found another morphotype that did not match the other ones that were already described. So we initiated a study where we investigated 19 clones and saw that we find a high phenotypic plasticity among the Coleps morphotypes, and that the algal endosymbionts were facultative and not obligate, which means that only from the color you cannot identify the species. Again, we found that the symbiont was Micractinium conductrix, and we also analyzed the nuclear ribosomal operon. What we found was not only the high phenotypic plasticity, also from these classical features of spines and wings and so on, that they were highly variable even within the clone cultures, but we also found two genetic groups. And we found one group that is now again called Coleps viridis, because we have heterotrophic species and we have mixotrophic species. We have also groups where we have heterotrophic and mixotrophic individuals in the clone cultures. And we have another group where we did not find endosymbionts so far, but there was only one strain that we investigated by ourselves. But anyway, we found that there were two groups that were only, and it was only possible to genetically identify those two groups. And finally, we now end up with Coleps viridis, where all the three former morphospecies are included. We have Coleps with and without algal endosymbionts. And the second group, Coleps hirtus, without the algal endosymbionts. And for the algae, again, we have this Micractinium conductrix. This belongs to the chlorella clade, and it was known as one endosymbiont also of Paramecium busaria. Then the next example I would like to show you is Urotricha. And Urotricha, here you have again the Oligotrichidae, the community of the planktonic ciliates, is one key genus. And there are about 40 described species, and they are very efficient algal feeders that dominate in spring in temperate lakes, and together with other ciliates and rotifers, they really graze down the phytoplankton spring bloom that is very typical for temperate lakes. So they are very efficient grazers during this time. But a taxonomic key to the larger species was available, but for the small species, there was no key available because they are really hard to identify from morphology only. There was only a key for the larger species available. And the larger species, they normally have 3 or more caudal cilia. So this is a species with only one caudal cilium, and these ciliates use the caudal cilia for a jumping movement. Just as a short notice. And what else was not available were molecular sequences. So even if you have this important ciliate group in the plankton, and you would have made a high throughput sequencing study, for example, none of them would have been named as an Urotricha sequence, maybe as environmental sequence or as uncultured ciliate. And this is why it is so important that you sequence ciliates that you identify from microscopy and that you can name finally. And again, we made some clone cultures of 6 of the small morpho species and also some morphometrics. And we also assigned our species to certain morphotypes, which was not easy because especially for these extremely small species, and small means about 15 micrometers in length, 20, they are really small. And so it's really hard to figure out the main features of these ciliates, even from microscopy. Then again, we sequenced the nucleoribosomal operon, and we also did some experimental approaches to figure out the perfect media or food for some of them. From the molecular phylogeny, we saw that we had certain species, but we investigated also different populations from different lakes, indicated by these differently colored dots. And what you can also see here, we again have cryptic species complexes also as we found for the Coleb species. Then another ciliate that is a keystone species in temperate lakes is Halteria grandinella. And here we also made some co-occurrence analysis and found, for example, that in the cold season, in the winter season, the species obviously is significantly correlated to many more food options, maybe, than in the summer months. But this might be just a matter of season, because in the cold season, of course, all the microbes are not such abundant as in summer, and the ciliate probably needs to rely on a high variety of food in comparison to summer, where there is the preferred food available in much higher abundance. But again, you need to know the ecology of this ciliate species and how it is also adapted to predators. And last but not least, and another example of which we also already heard a lot of things before, this is Paramecium busaria. And sometimes it's also worth taking a look on established model ciliates. And as you already saw, it's a mixed trophic ciliates that lives in symbiosis with green algae, but also a lot of endocytobionts can be found in Paramecium species or also Paramecium busaria. And this species is not a planktonic species. It prefers living on surfaces, on submerged leaves and stones, for example, and more in the shoreline of lakes or in ponds. Paramecium busaria is a model ciliate that is spread all over the labs over the world. And a model includes that the ciliate can easily be maintained in the lab, and that you can also manipulate the cell lines. So for example, that you have symbiotic ones and that you also get rid of the endosymbionts. This was also again shown before. And there are many strains available. But each lab has a focus on a certain research question, but we now aimed just to collect these lab strains and check them from morphology and molecular sequences. And again, here we found a high phenotypic plasticity, and indeed the phenotypic plasticity is really high. So here we have just some examples of the morphological features. But what you can see here is that also the size of these ciliates can be really very different even in the same culture. Then what again we found was cryptic species or syngens, even within the same habitat. Twice from the same habitat, we found different syngens in the same habitats. And again, we found various algal endosymbionts in the different cryptic species. So when we have a look onto these 5 syngens, now then we see that we have different endosymbionts in the different syngens, but the more we take a closer look also onto the algal endosymbionts, I'm sure that the one or the other symbiont will also show up, as for example for this syngen where already 3 different algal endosymbionts have been found. But for the other features, they are more or less all the same from morphology. And here is also again an overview onto Paramecium strains from the different syngens. They look more or less all the same. And here you have different strains from different origins with varying endosymbionts, and they belong to the different syngens. So these are marked with R1, 2, and so on. So again, we have cryptic species here. So to conclude, the state-of-the-art approaches, they need traditional approaches on the taxonomy and ecology, and of course also the molecular and bioinformatics tools, because then you have a very good integrative approach that can also be used for future water quality assessment, because when you then take a sample and also with the molecular approach for high throughput sequencing, eDNA, and so on, and you can figure out which species are indeed in this water sample, and from the knowledge on these ciliate species, you can then draw conclusions on the water quality. So what do you need to unravel ciliate out ecology? You first need a ciliate. You check it by morphology, single cell sequencing, create some co-occurrence networks, check out the symbionts, and draw conclusion on its out ecology that need to be verified also from experiments. And it's also important that you know more about the ciliate itself. What else is extremely important, and what also Ellen already impressively showed, is that you bring your protists into school classes and citizen science. I will not go into detail here because Ellen has impressively shown what is possible. And so again, which information is now necessary to understand the role of a single ciliate or protist species in its environment? It's the morphology, the molecular sequencing, the ecology. Where can you find it? How does it live? How does it move? How does it feed? On whom does it feed? Which are the predators? So, and I also recommend an interdisciplinary approach, because sometimes you need specialists on the endosymbionts, for example, or also on whatever is involved into your ciliate investigation. And finally, you can use these data for water quality assessment. Of course, I cannot do all of this work alone, so I have a very good co-occurrence network, and I'm happy also about all my collaborators. And last but not least, still many important and different species are out there. So I hope I could raise your interest for also all the other species. And thank you for your attention.
Thank you.
Thank you, ma'am. It was a wonderful talk. Any questions?
Thank you very much.
Can I have a question, please?
Ma'am, there's a question.
Yeah, Bettina, thank you very much for your talk. Can you hear me?
Yes,
Bettina?
I can hear you.
Thank you very much for a nice talk. I just wondered how often do you come across chlorella-free Bursaria in nature? And have you ever tried to infect them artificially with chlorella again? And is there any chance of a quick success in this enterprise?
I by myself did not find any chlorella-free Paramecium bursaria in the environment, but I know Sergey found some, and also I guess also Alexei found some. There are records of findings also from Japan. There is a record, but I myself did never find one. And we also did some experiments with chlorella-free Paramecium bursaria in the past and infected them with foreign chlorella strains. And when they had the choice from a mixture of different chlorella strains, finally they chose their original ones again. But they took also the others. So maybe if there was no chance to take their own ones again, they might keep also foreign ones, even from other ciliates.
From other ciliates. And how long did they keep them?
This I cannot remember anymore, but we have published this about 15 years ago. I can send you the publication. I cannot remember exactly for how long they kept them, but at least for some time so that we could see that they established a symbiosis.
A symbiosis, yeah. Thank you very much. Thank you.
So, ma'am, on my personal behalf and on behalf of the organizers, I extend a warm thank you to Professor Bettina Sontag. It was really amazing to see tortoise in a drop of water in the starting and then going ahead to understand the specific role of ciliates in the environment. And then when you establish the key organisms, ciliates as key organisms in aquatic environments and how they have been the most important consumers of algal biomass, and then quite amazingly then how they can be excellent model systems for addressing ecological as well as ecophysiological issues. And it's quite a roller coaster ride into the beautiful world of ciliates. And then how beautifully you have, devised that, give that highly diverse ciliates with different lifestyles and rendering them to different mode of nutrition, and then going ahead to, investigating them in traditional approach, and then going on to molecular as well as bioinformatic tools. It's been a wonderful, wonderful ride, ma'am. Thank you so much.
Thank you very much. Yeah, thank you for the invitation. Thank you very much.
We would like to hear more from you maybe next conference.
Yeah, it's my pleasure. Thank you very much.
Pleasure having you, ma'am. Thank you so much.
Thank you.
Thank you so much, Dr. Anita Narang and Dr. Gitika Kalra, for chairing the session, and to Professor Bettina for a wonderful talk. Moving over to our next talk, I would like to invite our co-chairs, Ms. Gauri Ghai from Department of Electronics, Acharya Narendra Dev College, and Dr. Anita Thakur from Department of Botany, Acharya Narendra Dev College, University of Delhi, to please introduce our chairpersons for the session.
Thank you so much, Rimpi. It gives me immense pleasure to introduce our session chair, Dr. Anupama Shukla. Dr. Anupama Shukla has done her undergraduates, masters, MPhil and PhD from University of Delhi. Her doctorate was in the field of mycology and was completed in 1995. She has been teaching in A&D College since 1995. She has been efficiently carrying out various responsibilities like being the governing body member, editor of college magazine, admission convener, etc. She is a life member of DUBS, Delhi University Botanical Society, and INSCAR. She has published 6 papers in international and national journals, chapters in books, and was instrumental in creating e-content for undergraduate students. She has organized several workshops at the college as well as for DBT-sponsored Star College program. I welcome you, ma'am. I invite Ms. Gauri Ghai for further proceedings. Over to you, Gauri.
Thank you, ma'am. I take immense pleasure in introducing Professor Anju Agarwal, who is a professor in the Department of Electronics with over 27 years of teaching experience at the undergraduate level. She has been a gold medalist in her MSc Electronic Science course at the University of Delhi. Her main area of research has been microelectronics, which includes modeling, characterization, and simulation of short channel HEMTs and MOSFETs. She has special interest in digital electronics, microprocessor, and embedded systems. She has been recognized by the University of Delhi for independent PhD guidance and has co-supervised a PhD thesis besides guiding more than 25 students for summer research. She has authored or co-authored more than 60 papers in peer-reviewed journals and international and national conferences proceedings. She continues to be a member of internationally acclaimed academic bodies, including IEEE and IEEE EDS Society. She has been a reviewer of IEEE transactions on electronic devices, besides being a member of Committee of Courses, University of Delhi. She has also served in capacity of jury member for national level exhibition and project competition, Inspire program, run under the aegis of Department of Science and Technology. She has also been a co-project investigator for Project Multipurpose Quadcopter, funded by National Science and Technology Entrepreneurship Development Board. She has also discharged her duty as an active member of organizing committees for International Symposium on Microwave and Optical Technology. I welcome you, ma'am.
Thank you, Gauri, for your kind introduction. I now take immense pleasure in welcoming Professor Cristina Missili. Professor Cristina Missili is presently working as a full professor at the School of Biosciences and Veterinary Medicine, University of Camerino, Italy, since 1993. Professor Cristina obtained her master's degree in biological sciences from University of Pisa in 1979, and thereafter finished her research fellowship, which is equivalent to PhD, from University of Pisa in 1983. In 1984, she started her career as a researcher in zoology at the Institute of Zoology, University of Camerino. From 1985 to 86, she worked as a postdoctoral research fellow at the University of California, Santa Barbara, USA. In 1987, she started her teaching career as an associate professor in zoology at the Department of Molecular, Cellular, and Animal Biology, University of Camerino. Professor Cristina has a long list of honors and awards to her credit. To list a few, She has been a member of the executive committee of the Italian Society of Protozoology several times and has been vice president of the International Society of Protozoology. In addition, Professor Christina has also been a member of the steering committee on ciliate molecular biology. Not only this, ma'am has also been co-organizer of several international conferences. Professor Christina has a number of academic nominations at the University of Camerino. She has been the director of the School of Advanced Studies, pro-rector for doctoral education, and scientific coordinator of the National Research Project for Antarctica on genomics and proteomics of ciliates. She has also been the principal investigator of the project on genetic tools to manipulate ciliates. Her research activities are primarily focused on molecular and cellular biology and ecology using eukaryotic microorganisms. Her study also involves ciliates as models for the response to environmental changes in temperature, salinity, and to other environmental stresses, toxicity tests, and gene expression studies. Without taking more time, I welcome you, ma'am.
Okay. Can you hear me?
Ma'am.
Yeah. Can you hear me now?
Yes, ma'am.
Okay. Okay, thank you. So thank you very much for this wonderful introduction, even too much. So now I try to share my presentation. Okay, here. Can you see my presentation?
Yes, yes, ma'am.
Okay. Now I put it in presentation. Is it fine? Can I start?
Definitely, ma'am, sure.
Okay, so thank you very much. So first of all, I would like to thank the organizers for giving me the opportunity to talk to this nice audience. And I changed the topic. I changed from the symbiosis that was so interesting in the previous presentation. And now I try to give you some information about the possibility to use ciliates as models to understand the response to environmental changes. So as you know, in the environment, there are many different conditions that change rapidly, and they produce stresses to the living organism. And we have different types of stresses. For example, we have natural changes in the environment, such as in the water. We have changes in salinity, changes in temperature that can happen for different reasons, like climate change also have this influence. Then we have external contaminants that are also problematic for the environment because they are substances that may affect the environment and affect the living organism. Ciliates are directly exposed to the environment in the water environment, and they have to produce some mechanism, activate some mechanism to cope with the changes in the environment. So in this sense, they can be considered good models. But how does the cell respond to environmental changes? Is it possible to study ciliates to understand the answer to this question? This is what I'm trying to show you, okay? We have, of course, to study at also morphological level, to study the phenotype of the ciliates, but then also we can study at molecular level, and we can apply some omics techniques. So means, of course, to have a reference genome when it's possible, to study which are the genes that are expressed for the response to the environment, and possibly when it's possible also to study the proteins, of course. And in this presentation, I will try to show you some examples that we have studied. One example will be with external contaminants, and then another one on natural change. And then I will show just a last research about another external contaminant different from the previous one. So let's me go on. We focus the attention on the analysis of the response of ciliates to silver nanoparticles. Why to silver nanoparticles? Well, actually, they are a common contaminant in the environment at present because they are used in a lot of industrial processes. They are used also in the health because they are biocides. So some materials of the hospitals use silver nanoparticles to protect against bacteria, and also cosmetics, for example, use some silver nanoparticles. Why they can be toxic? Of course, because there is silver that is a metal and is toxic, but also for their construction in nanoparticles, because if you see this figure, you see that the ratio between the surface and the volume increase with the size that became small. So if we have small nanoparticles can be between 1 to 100 nanometers. So if you have these small size particles, they have their surface exposed. Many of them can be present and they have a strong reactivity with the environment and with the organism. Why ciliate should be interesting in this sense? This actually was described in another paper before we started to work on this. And this picture is taken from the paper that is here cited, Bondarenko et al. You can see that you maybe not see very well because the picture is not so nice, but anyway, in this gray square here, there is a small tetrahymena, okay? And the authors tested different organisms with nanoparticles, and while some of them, like crustaceans, these crustaceans are very sensitive, actually tetrahymena appeared very resistant, able to activate some mechanism that may cope with this problem in the environment. And similar is also if we test only the silver. And the silver was tested by the authors because they wanted to understand if there was only the effect of the silver or also the effect of the nanoparticles. But actually this work does not arrive to characterize different processes that are involved. So this is what we try to do. We try to establish a procedure that can now be used also for other systems, not only silver, just the contamination by other metals or other type of stresses. So the procedure consists, of course, in set up the culture of ciliates, having the characterization of the substance that you want to test, and then you have to test how this substance is toxic. You have to analyze the cell, as it is shown here for tetrahymena and silver nanoparticles that can go inside the cell, and the cell can maintain this for quite a long time. And if, since we wanted to understand which processes were activated at molecular level, so which messenger RNA were isolated, which proteins were produced, so what we did was to isolate the RNA, and then we just synthesized the cDNA, and we tested by real-time PCR some genes that we know, just to be sure that the cDNA was well constructed, and also the isolation was giving some, isolation of RNA was giving some result. And then we sequenced completely everything that was selected. And then we did a bioinformatic analysis on the sequence of these transcriptomes materials. And we used a tool, a platform. The most friendly that I can for the moment suggest is this one called Omics Box. That is a platform that gives you the possibility to quantify the expression of the different genes that are expressed, or the different transcript, and by, of course, obtained by your analysis, and then by this quantification to select those that are differentially expressed, to understand which genes are going up, which genes are going down. Then with this same system, you can understand which are the proteins encoded. Of course, in tetramina, this is quite easy because tetramina is very well known in terms of genomics data. And so we can understand which can be the protein, and we can finally understand which can be the processes involved. All this has been already published, but I show you just as an example. And this is the first page of the publication. Okay, so let me show how we started. So we have to start with a toxicity test to understand how the substances are toxic. And in this case, you will see that the nanoparticles sometimes are also called color gold, because this is a name that is given chemically to the silver nanoparticles. So you can see this in pictures sometimes. Okay, so we tested the different concentration with cells and understand the viability of the cells, and the concentration were tested to find at first what is the EC50. What means EC50? Means that 50% of the cell die, the other remain alive. This means that this is the limit for the cell to survive. Then, of course, since we want to test some messenger RNA that may help the cell, so we took in consideration concentration that are less than the EC50, because we believe that the cell may have time to respond. And these were the concentration that have been tested. And then with the help of some chemistry department, actually this was done in collaboration with a center for nanoparticles that is in Estonia, in Tallinn, where they are particularly expert in evaluating them. And they gave us exactly the concentration of the silver that can be present in the environment released by the nanoparticle. And in a comparable experiment, we used silver nitrogen to have the same concentration of silver by this treatment that was released by the nanoparticles. Why this? Because we wanted to test if the toxic effect that we analyze is given only by the silver in the environment or by the nanoparticles due to their shape and their construction. So we did all the procedure with this, as I showed you before. The concentration were considered good for the isolation of the RNA, and then we did the test. When you do this kind of RNA sequencing, you have always to do at least in triplicate, because you know that there can be differences, and then you have to analyze at bioinformatic level statistically to see if there are differences. So this is a sort of graph. I don't know how many of you know the system. This is principal component analysis. So you cluster together together what is similar in the result. So you can see that the controls, actually we had 4 controls instead of 3 to be more sure. The controls are pretty close together, and the other treatments also form some clusters together. So this is a good indication to start. And then we analyzed which genes were upregulated or downregulated. These just show numbers, and some of these numbers are genes that are shared, as you can see in this picture here. Some are not shared, but the main problem in this is that, as you can see in the picture, we have many cases of still unknown function. So this means that there is still a lot to do in terms of finding the function of genes. And this is not only for Tetramina, it's also for many other organisms. But anyway, we have also a good number of genes for which the function is known. So, of course, the genes are many. So by the system, you have to try to analyze different groups that are considered to belong to specific processes in the cell. So, for example, for sure, in the case of silver, we expect a certain type of response to oxidative stress. And in fact, many of the genes that are listed here, they are by oxidative stress. However, if you see, this is what we call heat map, okay? The color show, if it's more pink, show higher expression. So, what was really significant, strongly increased by the treatment with silver in this group were mainly these 3 genes, while using the nanoparticles at the highest concentration that we tested, that was still much sublethal, we found many genes, many more genes that were significantly involved, so or upregulated or downregulated. So this work, of course, must be done in different processes, and it's pretty long, but at the end you get some result. First of all, to be sure that what you are searching is trustable, you need to validate at least, I don't know, usually in the papers we find 8 to 10 genes. You have to validate by PCR, quantitative PCR, to see, okay, what I calculated on the basis of my bioinformatic analysis is trustable. So you can do by chance, or you random, or you can do specifically. In this case, we selected some genes that we thought could be interesting for our purposes, such as metallothioneins, for example, that in Tetramina eliminate the metals, or others that we know. And we have the data here in the heat map related to the bioinformatics, but we also tested them by quantitative PCR. And if we plot the result by quantitative PCR against the results by the RNA sequencing, so by the bioinformatic analysis, and we have done this for many genes, each dot is 1 gene, and you see that they are in this line, so means that they are quantitatively very similar. So these validate our data. It means that the bioinformatic analysis and the RNA that was taken was actually in good condition for what we expected. So these encourage us to identify the processes that were involved. How can you identify processes? Not only looking at genes, but going into a very rich database that is called KEGG. I think many of you know it. And in KEGG, you find picture like this. Okay, this is improved by us, but anyway, for example, two of these proteins that are here are proteins related to the proteasome. So when they are upregulated, means that more proteasome is necessary in the cell and more degradation of protein is taken out. This is a very simple system. So studying these processes, we arrive to this general vision about the response of tetrahymena to the stress by nanoparticles compared also to the stress by simple silver nitrogen. First of all, it was clear that silver nitrogen enters forming pinocytotic vesicles that involve cluttering, not for the nanoparticles that form phagocytosis. This is the reason why they last for a long time. And this is also a problem in the environment for the bioaccumulation, because tetrahymena, of course, accumulate, and in the food chain, other organisms eat on tetrahymena and they take the big accumulation. And then we can see another very interesting aspect, at least in our vision, that is the activation of ion transporter molecules, channels. They can eliminate the toxic material, and they are shared. Okay, they are present in both cases, and this system actually works very well for the silver that is in solution, and work also for the nanoparticles, but since they are bigger, the phagocytosis inglobate them and go on in the process. What happens to the cell? The cell does not block the cell cycle. The cell tries to go on and to divide, and this is positive for the cell, and activate a very strong oxidative stress response by, how do you say, by upregulating many of these genes, much more than the genes that are upregulated by the simple silver that is in the medium. And of course, also some activation of mitochondria is necessary for the response. Of course, there is a strong peptidase activity and also the proteasome activity, as we have seen. So this is a sort of design of how the cell respond. And this is also summarized here. So we have, of course, the demonstration that this procedure function, tetrahymena can be studied. Silver nanoparticles also have an effect as nanoparticles. And of course, there are many other genes to be studied that can be involved. So this is a first example that, of course, could go on in the investigation, but at least give an idea. I want now to give you another example that is a natural change, and in this case is increase in salinity. Why we have increase in salinity? In many tropical environments, for example, we have increase in salinity during the tides that change. We can have increase in salinity for evaporation, increase in temperature. Some euplotes species were already known to form cysts in response to this. And you know, cysts are formed in many different organisms, including ciliates. Some cysts are very hard. Some other cysts are less hard and they last less time. This is our case. So we studied this Euplotes rariceta. It has been collected in Africa, Tanzania. It has been collected recently, but it has been also collected many years ago. Actually, I worked on this many years ago with Professor Luporini and Professor Dallai, and we identified this cyst and studied at the scanning microscope and electron microscopy. And we see that the cyst protect the cell for the time of the tide. When the tide come up again, the saline, because they live in small ponds in the rocks and where there is a little sand. And when the tide is low, there is low water, evaporation, high salinity, and they exist. When the tide go up, they exist. So this was studied at this morphological level, but we did a different study. I don't know if I'm able to show you this kind of little video. Yeah, you see, it's rapid. Encystment is very rapid, and the cell cover, you have here 5-second frame. So altogether is 3 minutes, okay? 3 minutes, cyst is formed and is covered, but also the cilia are all grouped inside and the nucleus is stopped in the activity, as we can see. What about the encystment? Encystment is also rapid. Actually, it's a little longer. This is 20-second frame. So it is about 5 minutes for this, okay, altogether. So we wanted to study this in the same way of the example of before, and we needed to have a system to induce the encystment easily. We could not simulate easily the tide, so we studied which was the inducer in the salinity. We tested calcium, magnesium, and sodium chlorides because they are all present in the seawater. And finally, we found that magnesium and calcium don't work at all, but sodium chloride, if you increase the salinity of the water by sodium chloride, as it is shown here, from 35 per thousand to 39 per thousand, you have a high formation of cyst, and in 1 hour you have many. Of course, it's not synchronous, so to have a good number you need time. It's fast for each cell but not synchronous, because the cell has to arrange the cell cycle, okay? And then so we decided to analyze 1 hour and 2 hour. So this is the same procedure, okay? I have already described one part. And for what concern the the RNA extraction, we decided to extract 1 hour and 2 hour, and of course the control, and then without control is without changing salinity, then the sequencing, then the analysis. In this case, it was more difficult because for this euplotes, we don't have the genome completely well annotated. So by the transcriptome, we had also to study the genes. And this is the reason why the manuscript is still in preparation. But anyway, we arrived to some results. First of all, number of genes that were involved that were significantly different. You can see them also here. This is, of course, done by a bioinformatic analysis, but to give you an idea, each small dot indicates one gene, and everything that is black means that it is very similar, not statistically different from the control and the sample that was stimulated for 1 hour with increasing salinity. All the colored dots indicate genes that are upregulated or downregulated. And each little line here indicate also one gene. Of course, you have the description of all of them in the bioinformatic analysis. And we see that in the controls, there are some genes that are normally highly expressed, some other that are shut off. But then in the other system, 1 hour or 2 hours, you have genes that are downregulated and genes that are highly expressed, and there is a change in this. So also in this case, we can study the process in which these genes are involved. Of course, microtubules, for example, microtubules movement, the cilia, because the cilia are blocked. Other detoxification system, many of different processes. We can also, in this case, verify the analysis, checking by real-time quantitative PCR if what you get is good, checking some genes, and these, as you have seen before, it fit. Then we try to understand what happens to the cell. And we understood at least these processes that are involved. First of all, although it is not a metal, it's just a salinity, the first response is the same of the oxidative stress. This is because the ciliate has these to, of course, eliminate the toxicity that can be done due to the stress. Then the microtubules, cytoskeleton is involved, not only the motility like the dynains, that start to be more expressed because there is a lot of transportation in the cell, but also the epiplasmin. Epiplasmin is material that is in the cortex, like the plateins, neoplates, similar to the intermediate filaments. And this is actually very upregulated because it may help to compact the cell. Then what was very interesting was to find one protein that was involved in the regulation of the motility, and it seems enough probably this protein that is downregulated to make the cilia to be blocked for a certain time. So this is what happens in 1 hour. 2 hours is the process go on a little bit, and we have one important aspect. The cell, since there is a toxicity, the cell should arrest the cell cycle, and this is what happens, okay, by upregulating these proteins. But the cell should not go in apoptosis because this is toxic, and so caspases are inhibited, okay, downregulated. Then the cells start to produce, activate proteins important for secretion. And we wanted to analyze the protein that is probably involved in the cell wall of the cyst. We found some similarity in a protein with another one found in dictyostelium, but we have not yet characterized this. So also in this case, by this procedure, we could understand how the cell respond, okay, although many other analyses should be done. Okay, now for last, if I still have a few minutes, I hope I have a few other minutes, I would like to show you something on which we are working at present. Of course, also the other things should go on, but this is a new project. This is related to Antarctica, and we have some ciliates that live in Antarctica. And this Antarctica, of course, is a global pollution sensor. Why? This is the title of this project. Why? Because in Antarctica, of course, the environment was not contaminated up to some years ago. But now, of course, due to the currents and the situation that we have in the environment, we have different contaminants that arrive there. And they, of course, affect the organism, and so the organism are good bioindicators, and this is the general title of this project to which we belong. So in this analysis, we, together with the other people involved in the project, we want to test contaminants that derive from plastics, because, you know, in the environment, they are a big problem. And we analyze bisphenol, that is a component of plastic that is also released by plastic in the water, and polystyrene nanoparticles, because plastic in the water gradually degrade and produce nanoplastics that are, of course, taken by phytoplankton, zooplankton, ciliates are in this part. Of course, they may accumulate and they may pass to the chain, to the food chain. So in this, that we have a low, we have not so many data so far. We studied the Antarctic species Euplotes focardi, for which we have investigated the macronuclear genome, so we have a good reference. And we also compare with Euplotes crassus that does not live in Antarctica. And if we test, here we have few data, but if we test the bisphenol, this compound in the water, we can find that Euplotes crassus at certain concentration, I don't go into details, but at a certain concentration they die, and we have a certain level in which 50% is survive. If we try the same with Euplotes focardi, we see that is more sensitive. So means that this Antarctic organism is more endangered by this kind of material. Then, if we test the nanoplastics, the situation is different, because crassus is, of course, they die at a certain concentration, but focardi, it doesn't die. It is actually able to, some of them die, but then the cell establish a certain condition. So we want to study this. How does it happen? At the beginning, we thought, let's see at the microscope if this is true. So bisphenol, 2 hours, 24 hours, this is Euplotes focardi. You see that vacuoles are formed, the cells suffer, higher concentration suffer even more. With the nanoplastics, doesn't suffer so much, actually maintain ciliary activity and get a big vacuoles inside. Crassus respond differently. Of course, is more resistant, so at higher concentration we have some effect, but even with the plastic nanoparticles, it really changes structure, is not able to form vacuoles, and it died. Okay, so why? We thought maybe is the temperature that is different, and we tested the temperature, and we saw that actually Euplotes crassus that is normally at 22, if it is taken at 4 degrees, it doesn't die. It doesn't divide, but it doesn't die. However, is even more resistant to the bisphenol. Okay, so the temperature does not help very much. So there is a point to summarize. Euplotes focardi is more sensitive to the exposure of bisphenol A, but Euplotes crassus is more resistant, and Euplotes crassus, even at low temperature, is more resistant, so it's not the temperature. And Euplotes focardi is able to accumulate the particles by endocytosis, while the other is apparently not able to do. Why? we have to study genes to understand. We have for the moment only a working hypothesis that I can show you. So we believe that in this system, the Antarctic desaturases that are enzymes that make the flexibility of the membrane in all organisms, but in Antarctic environment, Antarctic organisms, they are particularly active and they produce a high flexibility. This may be one reason why we have a higher membrane fluidity in Oplotes focardi, and for this reason it is at risk with plastic in the environment more than the other organism, but is more resistant to the nanoparticles. Why? This is the mechanism of endocytosis, exocytosis that happens in this Antarctic environment is very slow, and maybe other genes are involved. We still have to understand, but is probably this system that should be investigated. So for this, we are at this point, but we already believe that to have discovered the sensitivity of the Antarctic organism is a big problem for the Antarctic environment in general. And we would like to go on in this investigation as we have done in the other cases. So I conclude here. I thank the people of my lab and also other collaborations, but I would like particularly to thank the people of the laboratory. Angela Pierzanti is the postdoctoral in my lab that investigate most of these aspects. Matteo Mozzicafreddo is a bioinformatician, and now we moved to another university, but still collaborating with us. Sandra Pucciarelli is a member of this group, although she is now quite independent, and she also collaborates. And Alberto is a new one that has also bioinformatic capacities, and so can help us as Matteo did. And Aristide is a PhD that does another project. So thank you very much. Thank you for your attention.
On behalf of the 3rd International Symposium on Ciliate Biology, I would like to extend my heartiest gratitude for the enlightening talk through which we have come to know how the ciliates are able to manage or cope up with the stress of toxicity via the AGNPs, BPAs, PSNPs, and so on, and their response to salinity. Thank you so much for the enlightening talk. Now, due to lack of time, I will forward any questions that we have to you by mail. So the co-anchors, please take up.
I see that there is one hand raised, so maybe there is a question.
Yes, if possible. Even 2 questions, but very quick ones. Christina, thank you very much. That's Elena from St. Petersburg. Christina, I wanted to ask you about is anything known about the lipid composition of Arctic ciliates?
Yeah, hello, Gori. Gori.
Okay. Okay. Can I answer? Can I answer?
Yeah.
Yes. Okay, about the lipid composition, okay, not so much in Euplotes focardi. This is a new approach. We have to find a collaboration for studying lipids, and but we know something about the desaturases that are present there, okay? So the enzymes that make possible to have more unsaturated phospholipids, okay? So we have analyzed them in the genome of Euplotes focardi and in comparison with Euplotes crassus, and also we used to identify this desaturase group, one desaturase that was found in one Antarctic alga in which this flexibility has been investigated of the Chlamydomonas group, but is living in Antarctica. And we found many forms of these desaturases in our genome. And now we want to search how they are expressed, if they increase in condition in which we have some stress. So this is what we know. About the lipid, I know that there is a group in Argentina, you maybe know Nussblatt. He studied lipids in the trimena, phospholipids. And in fact, I contacted him to have some suggestion to manage ciliates for lipid studies. Thank you.
Thank you very much. Thank you.
Thank you so much, ma'am, for giving us your valuable time. The anchors, please take over now.
Thank you so much, ma'am.
Thank you.
I thank the chairperson and Professor Christina for giving her time. With this, we have completed our talks for the day, and we will now like to move over to the poster presentation session. We have small videos prepared for all the poster presentations that have been submitted for this particular symposium. I would now request the admin to please.
Thank you very much. Thank you very much. So
thank you. Thank you so much, Professor Christina. Thank you.
Thank you. I hope we can stay in contact and we can have, of course, exchange by email. Thank you.
Yeah,
So
sure, Professor Massili.
I stop my sharing. Okay.
Good afternoon. The topic of my poster is morphology and morphogenetic description of a new species of the genus Aponotohymena, which belongs to Celiophora oxytrichidae. This species was isolated from a water sample collected from a pond located in the Garden of Five Senses, Saket, Delhi, India. The water samples were collected in large beakers along with the roots of, and morphogenetic development was studied under the microscope. The size of the cell was 92 into 36 micrometer. Undulating membranes were in typical Notohymena pattern. They had 7 to 8 caudal cirri, and the granules were present, which were yellow to green in color and distributed throughout the cells. The genus Notohymena was first erected by Blatterer and Feusner in 1988 on the basis of flexible body 18 FVT UM-Sim Notohymena pattern. However, later on, this genus was further divided into two genera, the original Notohymena and the Aponotohymena by Feusner in 2016. The species of Aponotohymena are they show a splitting of third dorsal kinety and they have 7 to 8 caudal cirri. The present species under the investigation, it belongs to this genus only on the basis of all the features which belong to the genus Aponotohymena. The species was compared with the different species of this genus. For example, Aponotohymena, Aponotohymena australis, N australis combination novo, N australis was 2008, N australis who, and N as ISO australis Gupta et al. 2017. On the basis of when we compare this species with different species, it has been found that this species was a new species on the basis of its peculiar body length to width ratio 1 to 10 micronuclei, color and distribution pattern of the granules. So this species is described as a new species.
For the International Symposium on Ciliate Biology 2022.
The poster's title is An Investigation into the Bacterial Prey Selection Habits Among Ciliates and its Implication on Optimizing their Cultivation Strategies in the Laboratory. Bacterial communities are often, numbers are often affected by the presence of or are regulated by the presence of protozoan flora. Now, even among bacteria, protozoans prefer specific bacteria, that is, prey selection is seen. This is seen in a study where the ciliate Tetrahymena and Chilodonella showed a preference for biofilm forming bacteria, Pseudomonas constantini or Serratia plymoutheca. Now, similar things are being sought after in our study where we are trying to determine the prey preference of bacteria of ciliates for specific bacteria, and if these providing these bacteria in culture will improve their growth in contrast to the traditional method where competing flora is present. This method involves the method of to determine prey selection in our case involves the after this can be verified using the 16S rRNA sequencing, which will prove the species identity for sure. And the same thing goes for the same identification process must be performed for ciliates, where the morphological identification using polygon and protarachal tuning will give their will give a genera level identification, and spore species level will identify it using the 18S rRNA genes. Now now these identified ciliate and bacterial species are grown together in a plate such that combination of 5 bacteria which be contrasted to the case of the medium of involving cabbage, and thus we can see where more cells will be observed in cabbage or in enriched bacterial cultures by the CFU counts, by performing CFU counts. So what are the applications of this study? If the specific prey selection of the species is not, if ciliates of the presentation.
Thank you so much for the poster presentations. Due to shortage of time, rest of the posters will be put up on the college website, and the results will be announced on a later date. Now moving over to our next valedictorian session, I would like to invite Professor Ravi Tuteja, Principal of Acharya Narendra Dev College. He currently working in Department of Zoology, Acharya Narendra Dev College, University of Delhi. He expertises in tumor immunology and molecular parasitology and is working on the biodiversity of ciliates and stress response in the ciliates. Dr. Teja has also authored a book titled Textbook of Biotechnology and a Cell Biology Practical Manual as well. Besides, he has authored a number of chapters for INNN and NSTL. He is life member of Indian Parasitology and a member of the Association of Teachers in Biological Sciences. He is awarded with Meritorious Teachers Award in 2017 by Government of Delhi. Thank you so much, sir, for patroning this symposium, and please give.
Yeah. Thank you, Rempe. Thank you so much for such a generous introduction. And I must be saying that I have been given very tough job of giving a vote of thanks, but I take it as my privilege to propose a vote of thanks and acknowledge the contribution of those who worked really hard in organizing this symposium, and that too online. On behalf of Acharya Narendra Dev College and Maitreyi College, University of Delhi, we express our sincere gratitude to all the speakers for gracing the symposium and sparing their valuable time. Thank you, all the speakers. This online symposium aimed to provide a platform for the ciliate interest group to exchange information, present the latest research findings, and establish collaborations to enthuse a large number of Indian and foreign undergraduate and postgraduate students to take up ciliate protists as their preferred research area, and to provide networking opportunities for students wishing to take up doctoral or postdoctoral research on ciliates. And we are sure that the symposium successfully delivered these objectives. We would like to take this opportunity to place on record hearty thanks to our collaborators, namely Maitreyi College and Indian Network of Soil Contamination and Research, for the support and guidance extended to all of us. We thank all our students, fellow colleagues, and non-teaching staff of the college for organizing this symposium. We would also like to thank all those silent members who prefer not to be in the front, but were there throughout the preparations. Last but not the least, we thank all the lovely audience for joining us today. In the last, I would like to request all of you to switch on your cameras so that we can have one photograph. Thank you very much once again. Requesting all of you to please switch on your cameras. अंजू मैडम आपका हुआ नहीं नहीं आपने कहीं पे अपना वो खोला होगा ना हाँ if Dr. Valentina is there, Emma sir, Elena, Professor Elena, Bettina also. Bettina, Professor Bettina. Okay, thank you everyone. Thank you, Komal ma'am. Namaskar. Namaskar.